Hello everyone, we are Group 7. We'll talk about Tlr4. What's Tlr? TLRs are receptors that recognize pathogens in the innate immune system. In this video, we will take a closer look at one of these receptors, Tlr4. Before we do that, let's briefly review the initiation phase of the innate immune response. In body, immune cells are constantly monitoring for the entry of foreign substances. When a pathogenic microbe invades, the immune cells roughly recognize it with their sensors and declare a state of emergency. Someone is invading! What kind of system enables this rapid recognition and declaration of emergency? Pathogenic microorganisms have a common structural pattern for each type. Macrophages have receptors on the cell membranes that recognize this pattern. When these bind together, the macrophage recognizes the foreign substance. This receptor is called a pattern recognition receptor, and the ligand is called a pathogen-associated molecular pattern. They are the essence of the sensor. There are various types of PLRs, and each PLR recognizes a rough pattern of pathogens. For example, toll-like receptors, C-lectin receptors, knot-like receptors, and rig-like 1 receptors. There are some types of TLR. Let's introduce them according to the difference in their binding partners. First, there is Tlr4, which recognizes lipopolysaccharide, a component of the outer membrane of gram-negative bacteria. Next, there is Tlr1, 2, and 6, which recognize peptide glycan, a component of the cell wall of gram-positive bacteria. Next, there is Tlr5, which recognizes flagellin, a component of flagella. Finally, there are Tlr3, 7, 8, and 9. These are found on the endosome membrane and recognize nucleic acids. The presence of nucleic acids in the endosome means that a virus is present. How does the TLR binding cells declare an emergency? For example, when Tlr4 recognizes LPS, immune cells that recognize gram-negative bacteria release inflammatory cytokines, lipid mediators, and chemokines. Inflammatory cytokines and lipid mediators act on vascular endothelial cells and widening the endocytic gap and increasing vessel diameter. Chemokines attract leukocytes. In other words, they promote an immune response by leukocytes. Let's look at how immune cells release cytokines. When it recognizes LPS, Tlr4 forms a dimer. MYD88 and MAL are adapter protein. They combine the TLR domain of the TLR and RR complex. The RR complex recruits TLR6 and TRK1 complex. TLR6 gets polyubiquitinated and binds to TAB1, 2, and TAC1. Next, the RR complex phosphorylates TAC1, which leaves the polyubiquitin chain. I-kappa-B kinase binds to the polyubiquitin chain, and IKK gets phosphorylated and activated by TAK1. Activated IKK phosphorylates I-kappa-B. This activates the transcription factor NF-kappa-B, which I-kappa-B binds to. NF-kappa-B goes into the nucleus and promotes the transcription of cytokine genes as a transcription factor. This is a process from the binding of LPS to Tlr4 to the release of cytokines.